My name is Anna Morris. I am an HLA co-director at Emory, and I'm here on behalf of our group that includes Dr. Howie Gable, Dr. Nat Tambur, and Dr. Sebastian Height to talk about where we're going with epitopes, eplets, and transplantation. Quo Vadis. I don't have any disclosures. So as a brief way of introduction, as clinicians, you've likely been inundated with matching algorithms the last 5 to 10 years, and they each seek to understand whether we can identify low-risk transplant pairs and possibly minimize immunosuppression. And one major avenue has been through eplet matching or molecular mismatch load analysis. And so STAR mentioned this concept of molecular mismatch load back in 2019. And as you can see, in 2019, there was, we identified the gap that we needed tools to prospectively assess, prospectively determine immunogenicity beyond just the molecular mismatch load. And then in 2022, we approached the subject again and found that the molecular mismatch load does fall short of a biomarker because it lacks rigid thresholds. And so using it as a biomarker is not feasible. And the gap at that time remained that the need to better understand immunogenicity for assigning permissible versus non-permissible mismatches remained. And so what our group did was took a historic view of the literature. We wanted to redefine and take a fresh look at the context of antibody antigen recognition and hopefully provide some new information from our basic understanding of these interactions for epitope, eplet, and immunogenicity. And then we provide some clinical and research recommendations as well. And so we're going to use Slido here. And so there is a QR code on the screen that you can see. And so go ahead and take a picture of it and log in and help us provide some audience input for the question, what is an epitope? And hopefully you should see some of the answers supplied as multiple choice. And so what is an epitope? Is it A, a small region of non-self amino acids, B, 2 to 5 non-self amino acids within 3 to 3.5 angstroms? Is it C, bound by a paratope, C, immunogenic, E, helpful for matching, F, more than one of the above, and then G, none of the above? So I'll give you a few seconds. Is it working back there? It's working? Okay. All right, let's see. Oh, so I have to look at the screen so I can see the result here. Okay. We're seeing a diversity of answers, a lot more than one. Are you changing your answers? You're seeing everyone input? You're changing what you're doing? No. This is good. Thank you for your response here. We do see a variety of answers, but most of them are circulating on more than one of these. Well, I want to tell you, let's go on to the next question, then we'll come back to the answers. I clicked too soon, but we'll go back and talk about each of them. Okay, now what is an eplet? So similar answers as before, a small region of non-self amino acids, 2 to 5 non-self amino acids within 3 to 3.5 angstroms. Is it bound by the CDR3? Is it the immunogenic part of an epitope? Is it helpful for matching? Is it more than one of the above? Is it none of the above? Okay, I'm seeing more answers for non-self amino acids within 3 to 3.5 angstroms, but still a dominating answer of more than one of the above. So I want to go back here. I'll click this slide to go back to what is an epitope. And I want to get us all to the point where we understand that the correct answer here is it's the entire contact region of an antibody that's bound to an HLA antigen. So it's contact between the paratope and the epitope. And so what is an epitope? It is bound by the paratope. Answer C there for this one. And then if we think about what is an eplet, this was a term put forward by René Duquesnoy over the last 20 years to describe the potential target of an antibody within an epitope. And so if you put more than one, that's pretty accurate. All of these definitions have been used in the literature for what an eplet is. I think the one that's we like to center on mostly is B here, 2 to 5 non-self amino acids within 3 to 3.5 angstroms. But all of these, if you said more than one, they've been used in the literature before. And so as we thought about what antigen recognition is and how to explain this to the group here, we thought that facial recognition software was actually a good way to describe it, because there's many parallels to antigen recognition, and we use this on a day-to-day basis. And so what it does is it take measurements between these facial landmarks that allow for unique code to be generated for each individual. And so when you go to unlock your phone, when you set it up, you'd already made your code, and then when you go to unlock your phone, it checks to make sure that that code is the same. And so when we think about facial recognition, there's many woes. We all went through the COVID-19 pandemic, and we were dealing with wearing masks all the time. And for like a year or two of that pandemic, none of us could get access to our phone. We had to use our passcode. And so now it's much better. I was on the plane yesterday and wore my mask, and I could get into my phone. It was fine. But back in the day, we couldn't with our mask on. And even when we go out to the beach and we're wearing our sunglasses, it's still pretty difficult to get access to our phone. And that's because all of those landmarks are critical for recognition of your face. And if they're not all met, then there's no recognition. And so that's critical for the specificity, so that your sibling with your eyes cannot access your phone, and then your parent or child with your smile cannot access your phone. So when we think about antigen recognition to an antibody, the antibody there is in yellow and blue, bound to the antigen in green. If we peel those apart, those red amino acids are the contact amino acids between the paratope and the epitope of the antibody and the antigen. And so many areas are critical for recognition, and if not all are met, then recognition is compromised. And that includes complementarity, chemical contribution. And so if we copy and paste only certain regions onto another antigen, it's likely not going to be sufficient for recognition. And so the status quo for HLA antibody recognition for the last 20 years has been built by René Duquesnoy. It was a novel, paradigm-shifting idea of polymorphisms that they could be assessed in triplets, and sharing of these triplets between a donor and recipient, despite HLA antigen mismatching, could identify compatible pairs. And so that could be used for highly sensitized candidates to identify whether a certain donor would be compatible based on matching at this triplet level. And so that was HLA Matchmaker, but we, as is human nature, we wanted this tool to do more. And so it morphed into a prediction tool for antibody production. So instead of assessing which triplets were shared, now we're looking at these triplets that evolved into eplets to understand whether we're looking at antibody responses that have been developed against them. And so with this new vocabulary, with this came a new vocabulary for B-cell epitopes based on these hypotheses. And this new vocabulary differed from the original B-cell epitope literature, and that's for somewhat good reason. The B-cell epitope literature had been built by our understanding of viral and bacterial proteins. And those are completely different than self. However, transplantation of foreign HLA results in very minor differences than self. So if we look at this HLA molecule A1, those highlighted amino acids are those that are in A2 that are different from A1. And so you can see that now we're able to, when we study HLA, we're able to ask very specific questions about what is being recognized by the B cell. And so now we can ask questions like what amino acids are critical for binding, which amino acids are critical for inducing immune response, can we identify acceptable mismatches? But as we did that, assumptions were made, and the epitopes could be subdivided into these particular regions with particular functions and size limitations, and these assumptions were not based on previously known characteristics of antigen-antibody reactions. And so we would like to posit that when we make these assumptions with lack of evidence, it may lead to pitfalls in our analysis. So if we over and underinterpret an antibody response based on Eplets, that could be problematic. And then if we, determine that a particular Eplet is immunogenic, we might say it's immunogenic in everyone, when that might not be the case. And so to dive into this first assumption, a beautiful example was given in this recent paper from 2022, in which all 31 FYY-bearing alleles are to the left of that red line. But to the left of that line, you can see that there's a range of MFIs, with half being very high, the other half decreasing drastically in intensity. And so overinterpreting this antibody response could lead to the assumption that 31 FYY was a major target of the antibody, and so all specificities bearing it, despite low MFI below the threshold, should be called as unacceptable. This could be akin to throwing out the baby with the bathwater, or might be tossing out antigens, calling them unacceptable when there really is no need. On the other hand, underinterpreting the antibody response here could be assuming that the 31 FYY Eplet, due to the various MFI levels, it can't be the target of the immune response. And instead, possibly you only call the highest MFI specificities, or the worst case scenario would be you say this is non-specific binding and you call this nothing is unacceptable and you have potential to cross a very real DSA. So when we assess the amino acid polymorphisms across these HLA specificities that bear 31 FYY and maybe those that are reactive but actually don't have 31 FYY, we can see that it becomes clear that polymorphisms outside of that Eplet itself are capable of altering the binding of the antibody and then lowering the MFI intensity on the single antigen beads. So overinterpreting could lead to blocking antigens that are unnecessary, and underinterpreting could lead to crossing very real antibody. And so the next assumption, immunogenic in one, means immunogenic in all. So as you can see in this graph on the bottom, all antigens listed have the same epitope, but they cluster in groups with regard to their electrostatic potential in comparison to self, with two major groups emerging. I know it's kind of small, but essentially what this data was showing was that one mismatch in an individual with a certain HLA molecule may not carry the same risk compared to someone else with that Eplet and another HLA molecule. And so those physiochemical properties of the mismatch may differ in the setting of different HLA alleles, and then differences to self HLA will also influence whether that Eplet is seen as immunogenic. And so, because we can have some of these assumptions that can lead to clinical decision making, that can lead to pitfalls, our group wanted to go back to the basics and to understand and reestablish the foundation for how we move forward with understanding antigen recognition and antibody formation. And so what do we know about the B-cell epitope? Crystallized structures are the preferred way to study B-cell epitopes. In the 1990s, there was only a handful known, and that's what you see on the left-hand side. And that's why I showed you earlier, you have an antibody in yellow and blue and the antigen in green, and about 18 to 22 amino acids participated in both the paratope and the epitope, covering about a 900 angstrom radius. And now we have over 800 crystal structures of paratopes and epitopes, and about the same numbers are coming out from that data. 18 to 19 amino acids are averaging on both the epitope and the paratope, and both contribute to complementarity. Both contribute hydrogen bonds, salt links, hydrophobic interactions, et cetera, that allow complementarity to take place between the two. And on the paratope itself, 90% of those residues that are interacting are within the complementary determining regions, 1 to 3. And it's not just CDR3, which we like to talk about, but it's also the CDRs 1 and 2. About 60% of those amino acids are making contact to the antigen. And a concept that we've talked about with epitopes since the 1960s is immunogenicity, or the ability to generate an immunological response. And so what do we know about immunogenicity? First, factors that are innate to the individual influence the B and C T cell repertoires to recognize non-self. And so that includes HLA genotype. It also includes previous immunological experience. And so that creates the repertoire that will be able to recognize non-self. non-self. And then when we think in terms of non-self molecules, mismatched amino acids within that non-self molecule is contributing to shape and chemical complementarity, but they also can affect the surrounding amino acids that are beside those mismatched amino acids, and that can also affect the energetics of binding of the non-self molecule to the BCR or the TCR. Further, those mismatched amino acids in non-self also contribute to peptides that are able to be presented to T cells to provide T cell help. And so all of that is necessary. And then you think on the side of the B cell and the T cell, they're having a BCR and TCR that are able to recognize that non-self molecule with the mismatched amino acids. And all of that is going to be seen within the context of that orange circle here, a sense of danger, how much of the non-self molecule is present, the strength of the exposure, and also, since we're in transplant, the type and dose of immunosuppression. And so all of those factors will influence how a non-self molecule is seen by the immune system and whether it's able to stimulate an immune response. And so after that recognition, what's going to happen is I have a little tornado up here. You have a germinal center reaction that induces somatic hypermutation. You get a refinement of that antibody now, and that now has stronger affinity to your non-self molecule. And so those mismatched amino acids now of the epitope are going to contribute shape and chemical complementarity and also affect those surrounding amino acids too, which can alter the energetics of binding. And then on the side of the antibody, you also have amino acids that are participating in the shape and chemical complementarity. And so, immunogenicity then is not just based on foreignness or those mismatched amino acids, and we've taken really a zoomed-in approach when we study HLA eplets in particular. And so if we zoom out to look at the entire epitope, and its contribution to paratope recognition and stimulation of our B cells and our T cells, and you really understand those biological properties that then act on it, that allow for a more accurate view of the gaps that we face in understanding HLA recognition and development of donor-specific antibody. And so concepts that we might have put on the back burner, like HLA genotype, now that's very critical. And we also want to understand those surrounding amino acids that contribute either shape or chemical complementarity. And so those could be amino acids contributed to by the peptide or by the DR, DQ, or DP alpha chain. And so all of those, if they're contributing amino acids within that paratope region, they can affect immunogenicity. And so moving forward, an epitope now considers the entire contact region of an antibody that's bound to an HLA antigen. So again, we're talking about an epitope to paratope interaction, that entire contact region. When we think about amino acid mismatches that are shared by one or more HLA antigens, that can provide important binding energetics for the antibody, but other amino acids in the epitope are also contributing and are likely critical to antibody recognition. And it does not mean that those polymorphic amino acids, those mismatched amino acids, are in the center, quote unquote, of the epitope. And then we think about immunogenicity. It's contributed to by amino acid mismatches, of course, but it's also their effect on the surrounding amino acids, as well as all of these other biological properties on the system, including peptide presentation, T and B cell repertoire, et cetera. And so we cannot, let's see. It cannot be defined by single amino acid mismatches or eplets as we move forward. And it cannot be extrapolated to the population without high-resolution typing information as well. And then lastly, the single-antigen bead reactivity we read out with our HLA assays, it's really a measure of antigenicity. So what's taking place after that germinal center reaction, after we've stimulated a high-affinity antibody, but it's not reading out all of those factors that took place to create an immunogenic response. And so at this juncture, the molecular mismatch analysis provides one measure of immunogenicity. It's a blunt tool. It provides a count that is associated with risk of outcomes. However, those thresholds are not stringent, and it cannot be used as a biomarker. And right now, it doesn't consider the complex interaction or contribution of that entire epitope on immunogenicity. And so using it in isolation is not recommended. However, these tools can all be refined and improved to better understand, above and beyond account, but the hierarchy of these mismatches and where they land, which using the background of the immunology that we've talked about today. And so Eplet analysis is one of these tools of many to hypothesize antibody production and reactivity. And so we want to make a few recommendations for research for how to move forward. And so there's descriptive information to provide, which includes accessibility or structural analysis via crystal structures and or 3D modeling, high resolution full typing of our donor and recipient, amino acid assessment, whether that's alignment, but also looking at the categorical match, say like between non-polar, polar, acidic, basic residues, that type, and then also thinking about electrostatic properties. So those EMS 3D tools that allow for us to assess, okay, what is the electrostatic potential of this particular epitope? In addition to including that information, also providing evidence of an immune response. So thinking about, yes, single antigen bead assays, but also providing absorption and elution assays, human monoclonal antibody testing. We also have site-directed mutagenesis and other T and B cell assays that can contribute to providing evidence for an immune response. And then also looking at the evidence of the clinical impact. So thinking about cohort data with graft outcomes that can help us identify, okay, a hierarchy of potential mismatches. But again, all of that descriptive information is going to be important and necessary for those cohorts. And two, I haven't mentioned, and the potential gravity of using machine learning in some of these models going forward. Because of the complexity of the immune response and building it out, we can see that, in the future there could be avenues that are beyond our ability to assess that could be seen with machine learning, add to these tools to make them more robust. And so that was some feedback that we got from our webinar. And so the final recommendations are to get the international community involved to help us establish accurate terminology for these complex biological states and appropriate avenues for international participation. And then to have an avenue to publish these new antibody-verified mismatches and polymorphisms based on clearly defined levels of evidence. And with that, I'll conclude the summary of our working group on behalf of Dr. Gable, Dr. Height, and Dr. Tambor, and take your questions and your feedback. Thank you so much.
Anna, we're open for questions.
Hi, I would like to thank you for that wonderful presentation. I always learn something from your talk. I wanted to say something about the use of machine learning, which is imperative that we start using like bigger models and more like impactful data-driven strategies to understand these complexities. But then I feel like there's always this problem, which is the rebuttal that it's never been proven with an antibody, And we kind of have this problem all around. We are advancing in new ways and yet still stuck with our rebuttals. So what do we do about that?
I do think these tools can help. I don't think they're the final answer, or will never be, like machine learning. That I don't think it's going to identify, we'll still have to be able to prove that an antibody exists and is causing damage, right? But I think it may be helpful in the sense that it can provide things that maybe with the right cohort data we might not have seen before, and analyze, similar to some of the talks this morning. Like we have cohorts with many variables, right? And so it's hard to parse apart the effect of each of those on our patient cohort. So it might be where it can help us in these situations, but the need for robust cohort data to input into that machine, into those like mechanisms is paramount. Like we can't move forward with subpar data and expect to get answers out that are worthy to look at.
I want to challenge Michelle. Your assumption that it's imperative to use machine learning.
Thank you.
Why do we have that thought? In my mind, a machine can answer a question that we know how to delineate, and then it can quickly give us the answer, sift through a lot of data. But if we don't know how to phrase the question, I don't know that any machine can ask it for us and therefore may not be able to answer it. So I want to posit here to the whole audience. There's that thought that, oh, machine learning, and that would be the solution. But I think we need to be really careful thinking about those things.
Yeah, I agree. It's only as good as the data you have to put into it.
No, it's only as good
Garbage
as the
in, garbage out.
question you ask.
The question you ask.
Right? And who's going to ask the question? Is it the machine or the person behind the machine? I just want everyone to pause and think about it a little bit, because we're all following this huge trend, and sorry, but I'm doing this all the time, right? Let's stop a minute and think about what we're thinking. Let's figure out the question, and then we can give someone else or something else the ability to help us with this. I'm not sure that we know even how to ask the question, right?
And right now, do we even have enough data to put in a model like that to give us meaningful information? Yeah.
I don't think so.
No.
And a nice talk. In terms of the immunogenicity of Eplet mismatch can be differed by the patient's sensitization, previous sensitization histories, like a woman with multiple pregnancy or retransplant candidates can be entirely different when you compare with unsensitized patients. So how do you capture that in your research proposals?
I thought a little bit about this in regards to whether like your immunological experience and how it affects your B and T cell repertoire. And I think the first place you have to start is accumulating the data, right? Having robust data so that you know, okay, for each of your patients, they've received they're up to date on their vaccines, they've received flu, they've received, their COVID vaccines, et cetera, they've had a notable experience for mono, and they've also had these 3 pregnancies and past transfusions. So I can see the need for data collection, and right now, we don't provide any of that. And the HLA lab sometimes gets some of that data. We get maybe transfusion and pregnancy, but vaccine and immunological experience, other than that, it tends to be not known. So I feel like that would be important to moving forward and understanding like those repertoires that then shape and respond to non-self, but we have to start with good data collection.
Chris.
Hi, thank you very much, and a great talk. I miss Howie, but you've clearly taken on the mantle and are doing an amazing job.
I miss Howie too.
You clearly have such a great handle on all of this complex information. I want to ask a question and maybe challenge the assumption that we can't use these as biomarkers. And the reason is, we don't actually have to understand something for it to be a biomarker. In fact, if I could show you that the number of eyelashes on the left eye predicted ABMR, we could use it as a biomarker. We don't have to understand it. And there's iterations in our understanding of all of the things that we currently use for biomarker, not the least of which is currently HLA antigen mismatch, which has all of the negative problems that you pointed out for Eplet mismatch, plus many more. So I think, at least in my view, I totally agree with everything that you said in terms of the limitations of where we are right now, but I do feel that it's a step forward from where we have been, and it doesn't mean we don't have further steps to make. It just means that we're making iterative gains. So that's just a comment.
Yeah, I think there might be two directions of comment back. So one is whether it can be a biomarker, quote unquote, defined by like the FDA. And then second would be using it as a tool that we can now to stratify risk, which the second half of your question, I think, sure, yes, we can use it to stratify risk. However, there are ways that we can refine these tools to better understand risk for each individual. Like on a population level, we can get these categories of risk, but yeah, on an individual basis, understanding risk. I think there's need for understanding that hierarchy of, okay, these certain amino acid mismatches are more potently immunogenic than others. So just refinement of these tools. And then the first half, I believe, the first half of your question, whether it can actually be used as a biomarker for FDA, I think because the threshold changes and it's not rigid, that's why, like on the books, technically it can't be used, quote unquote, as a biomarker, but yet it can be used to stratify risk. Is that am I understanding that correctly?
Yeah, well, I think, Chris, your group is the one that really moved the field forward a lot with providing a lot of data. I think there are a few things that, as Anna had mentioned, the fact that every time that we have a new iteration of the software or the Eplets registry and so on and so forth, the threshold moves. I think that is definitely a problem. I'm not sure if our clinicians, clinical colleagues appreciate that there are actually different softwares who are counting those Eplets differently. And I think all of those put a caveat into how can we use this clinically. This is definitely a biomarker. It's not telling us much about the immunology behind it. And I think this is something else that is going to be really important for everyone to appreciate. A biomarker and the immunology behind particular processes are two separate things. So personally, my hesitations about the use of Eplet mismatch load or any other biomarker is that it takes the field away from trying to understand the immunology. But I think your data is really strong, and you've showed it on multiple cohorts. But now we have a different iteration of the software and different thresholds. So how would you propose, or actually there are no thresholds to the best of my knowledge, I'm not following this very closely. How would you recommend the field to use this at this point?
Yeah, that's a loaded question. I think, well, I can tell you with 100% honesty, we're using it already and have used it for years in Manitoba because it's clear that it provides some additional information above just traditional antigen matching. It's certainly not perfect, but when you have both pieces of information in front of you, it seems silly to keep relying on the old information that we know is far less accurate. And we could talk about examples of, where two people have an antigen mismatch. One's got a single Eplet mismatch and the other one's got 25 Eplet mismatches. That probably means something at the immune system level. I think for sure it's a big problem right now that as time has gone on, more and more people have developed their own homemade software within their institutions, and we have different versions of the software, totally legitimate big problems. And so very important, if you're going to use a threshold that's been published, you got to use the method that's been published. That is for sure. The only additional comment I would make is I don't see immunogenicity and counting up the mismatches as mutually exclusive. I think they can both actually be in the final
Agree.
A point where we're hopefully all going to get to. I actually see them as complementary. I mean, it just makes logical sense to me that if you have mismatches and you also knew about the individual immunogenicity of those mismatches, that could easily be statistically incorporated.
That would be perfect.
Yeah.
Roy?
Hi, thanks. That was a really fabulous talk. So I think As a clinician, if you think about eplet mismatching, I think everyone's right. You guys may be using it in all your patients, and I totally, and your data is fabulous, and I totally believe everything, that it works for you. But the reality is, it needs to still be proven as a context of use, and there is a randomized controlled double-blind trial that's currently enrolling, which will hopefully resolve that one way or another. So I think ultimately that will show whether it works or doesn't work. And I think that that's what we have to wait for. Having said that, not only are you using it in Canada, but like the National Kidney Registry is all in on it. So there obviously are people that believe in it, and it's great. But we, as a transplant community, have to all understand that if it works, if it advances the field, that's great. Tacrolimus advanced the field over cyclosporin. It doesn't mean that we should just say, that's it, we have an immunosuppression now that works. We still are compelled to look for better ways. We still have to understand why, we still have to understand the immunobiology, and as science advances, whether it's through, the technology of machine learning, or whether it's more understanding the actual immunobiology, and maybe it is related to more than just eplets, then we have to be all willing to change in the future.
Thank you for bringing up the NKR. I've got nothing against the NKR, but I do have a problem with selling dreams to patients. We have several reports, some of them were already published at last ATC, that showed that the intended donor or the donor that stepped in as the intended donor for a patient was swapped with a different donor because of eplet mismatch load that was lower, and those patients went ahead and developed ABMR within the first year. One of them even lost the graft, and I was contacted because it was a DQ antibody, of course, but we were hearing those cases. So I think there's one thing from using eplet mismatch as one additional tool in your toolbox, and you add it to a lot of other things that you put together. And there is a big difference between this and using eplet as the one thing that you allocate organs based on. And I really would caution the field not to jump to this, because I don't think that we have sufficient information for that. And personally, I think there are additional issues associated with this, but I'm not going to go into this right now.
I think it highlights the need for the community to really, the terminology means something. When there are things like zero eplet mismatch, for those who might be less informed, they may equate that to a zero HLA mismatch, and they're very, very different. So just to disseminate that knowledge to the community. Any parting thoughts, either of you two, before we go to the next speaker?
Elaine?
Oh,
Just one conclusion, sitting here listening, I think we need standardization. Everybody's calling for everything that we've done here today is about standardization. We haven't said it here because it seems to have launched in the field independently of maybe some thoughts on how we should do this uniformly. So that's what I'm thinking, hearing this, that we should make some motions to think about standardization. I appreciate moving the field forward, but and collecting the data, but if we're all doing it differently, we will never understand this data. So I think there's a call to standardize, and maybe this is something, Chris, we have to be thinking about as we go forward. And I appreciate the database is changing. People do class 1, do class 2, they don't put them together. I mean, I've never heard of such a thing, so I've been concerned about it. High resolution, intermediate, people impute, they don't even know the race. It's impossible. We're always going to have errors in this model. So if we don't standardize something, we will never get it right. So that's my thinking.
Thank you for that, Elaine. And I echo this, but I want to add something else to this. Our immune system is so complicated, and there are so many different things, and I think Anna was so beautifully trying to put that together, that even if we get huge cohorts, it's going to be very difficult to tease out individual mismatches of patients, how they contribute to eventually ABMR or graft loss. We must have functional data beyond the pen and paper counting mismatches or something like this, and I think that's a critical component. We have to have functional data.